Hello and welcome to Effortless Mastery. Today we'll be doing a very exciting topic, yet challenging for a lot of medical students and paramedical students. That's going to be the chemistry of nucleic acids. The chemistry of nucleic acids. So what do we regard as nucleic acids and what is their structure all about? That's what we're doing today. Now, nucleic acids usually come in the form of nuclear proteins, so in the form of conjugated proteins. So there are different types of proteins. We have simple and conjugated. Conjugated is when I have a protein part and any other thing called prosthetic group. So when you look at this, nucleic acid in the human body usually come in the form of nucleoproteins. An example is chromatin. You still remember from biology, basic biology, that chromatin is that stuff that will make up the chromosome or that we find in the nucleus of every cell. So within the animal cell, there is a nucleus. Within the nucleus, there's a chromosome. And what makes up that chromosome is chromatin. The chromatin is actually a nucleoprotein. Now, these nucleoproteins, when broken down, have two parts. The nucleic acid, which is what we are learning today, and the protein. Usually, if I'm using chromatin as an example, the protein we have in the nuclear protein called chromatin is called histone, whereas the nucleic acid is usually DNA, if I'm dealing with chromatin. But in the human body generally, the nucleic acids we can have is either DNA or RNA. What's the full meaning? RNA is ribonucleic acid. And of course, the name is given because of the particular type of sugar we have in it. In ribonucleic acid, the particular type of sugar we have is called ribose. But when you come to deoxyribonucleic acid, the particular type of sugar we have there is going to be deoxyribose. I hope we are together. So RNA is ribonucleic acid. The other one is deoxyribonucleic acid. And it's just based off the kind of sugar you have within them. Now, when I break down, of course, the protein will be broken down to form amino acids. But if we break down our nucleic acid, the enzyme to break down the nucleic acid is called a nuclease. So that means if it was ribonucleic acid, it would be ribonuclease. If it was DNA, it would be deoxynuclease, deoxyribonuclease. Now, this deoxyribonuclease is a phosphodiesterase. I will explain this as we go on. And this is because when you look at it, a nucleic acid will be made up of numerous nucleotides joined together by a phosphodiester bond. So to break it and release a free nucleotide, what I use to break it is a nuclease or a phosphodiesterase. As we continue the topic, you are going to so enjoy it and see why this is so. Now, additionally, from my nucleotides, the nucleotides can either be AMP, that is adenosine monophosphate, GMP, CMP. GMP is guanosine monophosphate, cytidine monophosphate, and others like uridine monophosphate and even thymidine monophosphate. Now, for now, just know that no matter the nucleotide I'm forming, how did I get them? Nucleic acid was made up of numerous nucleotides linked by a phosphodiester bond. So what I use to break it is a nuclease or a phosphodiesterase. Now, after being broken and we have nucleotides, nucleotides have a pattern. They have a base, a sugar, and a phosphate arranged in this pattern. So you always have the sugar moiety being in the center. You have a base attached to carbon 1 prime. Usually when we label the sugar, remember I told you the sugar can either be ribose or phosphate. I will explain this as I go on. When we label the sugar, we label them with prime. So what is a nucleotide? A nucleotide will be having a base attached to a sugar, and that sugar is still attached to what? A phosphate. Now we come back here and we have different types of nucleotides. So for me to break down a nucleotide, I can get rid of the phosphates. When I take out the phosphates from the nucleotide, what I will now have will be a nucleoside, having just base and sulfur and the sugar. And the enzyme for it is called nucleotidase. Nucleotidase, or a phosphatase. What is a phosphatase? A phosphatase is an enzyme that usually adds water to a compound and then bring out phosphates. So I add water to a compound aimed at moving phosphates. So when I add water to a nucleotide and I remove the phosphates, what I'm forming is a nucleoside. And the names can either be adenosine, guanosine, cytidine, what have you. Just mention their names. Then we now come to the nucleoside themselves have a sugar and a base. So what I use to split them will now be an enzyme called nucleosidase, or another name we can call them a nucleoside phosphorylase. For now, I'll not go into too much details about it. Once our video on metabolism of nucleic acid is released, you rush on it and then I will explain more about this enzyme. But for now, we'll just tell the fact that this enzyme will act on nucleoside to give us a sugar and a base. This is sugar moiety and a base. And please, the kind of sugars we can have in nucleic acids are either ribose or deoxyribose. What's the difference? It's still the same pattern of structure. Ribose means you have 5 carbons in that structure. And then it forms the only difference is that for a ribose sugar, all carbons will be having OH. For the deoxyribose, usually on carbon 2, that carbon 2 will be deficient of oxygen. So that's why the name is called, in fact, the correct name will be 2-deoxyribose. That means it is like ribose. The only difference is that on carbon 2, it is deficient of oxygen. So we call it deoxyribose. Then for my nitrogenous bases, they can either be purines or pyrimidines. Who are the purines I can have? Okay, I should even start with pyrimidines. Who are the pyrimidines I can have? I have COT, acronym, cytosine, uracil, and thiamine. Cytosine is seen in both DNA and RNA. Cytosine is seen in both DNA and RNA. Uracil is seen only in RNA. Please, that should be noted. Uracil is seen only in RNA, whereas thiamine is seen only in who? DNA. So please take note. Cytosine is seen in both RNA and DNA. Uracil only RNA. Thiamine only DNA. And their structure is a 6-membered ring having 2 carbon and 4. They have a common structure that is a 6-membered ring having 2 carbons and 2 nitrogens, 2 nitrogen atom and 4 carbons. So when you look at it, you count it from here. This will be nitrogen 1, number 2, number 3, 4, 5, 6. I repeat that. The common pattern of structure for pyrimidines is that they have a 6-membered ring having 2 nitrogen atoms and 4 carbon atoms. Nitrogen atom, this one will be labeled number 1, number 2, number 3, number 4, number 5, number 6. This will be their common structure. Of course, they will differ in some things. That's why they are different. For example, cytosine, you'll be having a double bond to oxygen on carbon, on this number 2 carbon, and it will be having a single bond to NH3, that's NH2, which will be an amino group. So usually we'll call it, for cytosine, the common name we give it is 2-deoxy-4-aminopyrimidine. 2-deoxy-4-aminopyrimidine. But when I go to uracil, uracil will be similar to him, but it will be having double bonded to oxygen here, double bonded to oxygen here. The name will be called, for uracil, please, you can be taking note of this, 2-deoxy, that is all, we can just say 2,4-dioxy, 2,4-dioxy. So I'm having it both on number 2 and number 4, oxygen. But here I had oxygen here, but then I had amino group on the number 4. Then for thiamine is the same thing as uracil, just that on number 5 carbon, we are going to be having a methyl group. So all I'm trying to point out from here is that I have 3 people which are pyrimidines, having a similar pattern of structure, a 6-membered ring with 2 nitrogens and 4 carbons. They differ in different things. Cytosine labeled or named 2-deoxy or 2-oxy and then 4-amino. Uracil labeled 2,4-dioxy. That means they have oxygen on 2 places, in position 2 and in position 4. Thiamine 2,4-dioxy, then 5-methyl. That means the only difference between thiamine and uracil is that thiamine is actually a methyl uracil, a 5-methyl uracil. Please remember, uracil is seen in where? RNA. Thiamine is seen in DNA. That's all to know. For purines, their structure has a pyrimidine ring and another smaller attached ring called an imidazole ring. Imidazole was seen in histidine, that's in chemistry of proteins. When I make videos on that, you are going to enjoy yourself, trust me. Now, that will now be for my imidazole, but this is what purines look like. They are a 9-membered ring having 4 nitrogens in total and 5 carbon atoms. Now, the person labeled number 1 is this other nitrogen. Observe in this one, the nitrogen labeled number 1 was this one. Over here, this is your number 1, and this is your number 9. I do not want to go through the stress, but this 1, 2, 3, 4, 5, 6, 7, 8, 9. That's how they are numbered. Who are the purines I have? We have the 2 major purines, adenine and guanine, both found in both DNA and RNA. So that means adenine is in DNA, it's also in RNA. Guanine is in DNA and RNA. Hypoxanthine and xanthine are just derivatives we have along the way. From deamination. So that when we remove an amino group, when we remove an amino group from adenine and guanine, we end up forming either hypoxanthine or xanthine. Specifically, guanine will be forming xanthine, whereas adenine will be forming hypoxanthine. That's when you get rid of amino group. So the only difference between this phase 2 and them, these ones are just things we see during catabolism. That will be in my video on metabolism of nucleic acids. I will explain that better. So these are the purine and pyrimidines I have. Now the next thing to now know from what we have learned is that if these are my nitrogenous bases and we have showed their number, when nitrogenous bases combine with a sugar moiety, a base plus a sugar, what do we form? A nucleoside. But we need to know, how do we name these nucleosides? If it was a purine, the name will end with syn. If it was a purine. And the bond will be between, please remember I told you that the ribose sugar or deoxyribose sugar is labeled with 1', 2', 3', 4'. So the bond will always be between the 1' on sugar. And if it is in purines, the number 9 on the purine. Whereas for my pyrimidines, the special ending will be dine for pyrimidines, and then it will be between the 1' on the sugar and the 1 on the pyrimidine base. So observe something I had mentioned. When you are labeling this, your base, you number them with normal numbers, 1 to 9 or 1 to 6, based on pyrimidines or purines. But when it's now time to number your sugar, you number 1', 2', 3', and so on and so forth. So please observe something. Anytime it is a purine, if it is a purine like adenine plus a ribose, the name given will be adenosine. A guanine will now be guanosine. But if it was, so that's how we end the names, purines, adenosine, guanosine. But if it was a pyrimidine, it will now be cytidine, uridine, and thymidine. So the spellings, cytidine, cytidine, sorry, dine, uridine, uridine, thymidine, thymidine. So you realize that with pyrimidines, you always end with dines. But with purines, you end with syn. So that's how we name the nucleoside, which has a base and a sugar. Please, the bond is between, they will always be between 1' of sugar. 1' of sugar. But in purines, it will be with the number 9. This nitrogen here, that was number 9. But then with pyrimidines, it will be with the number 1. So that must be noted. All right, so that is just that for nucleosides. Then for nucleotides, your phosphate, so following the addition of once I have a nucleoside, the phosphate, please note, in a deoxyribose, that is DNA, in DNA, observe something. There is no oxygen on number 2. We have the nitrogenous base on number 1. The places that phosphates can enter is either number 3' or number 5'. Usually number 5 most times. but it's either 3' or 5'. But in RNA, phosphates can enter 3', 5', and can additionally enter 2'. But commonly, what we tend to see a lot is 3' and 5'. Okay, so that is that for that. So that's the bond, phosphates. So once you have the phosphate added, that's going to be, you have your nucleotide. Now, what links many nucleotides together in a DNA or an RNA? What will link many nucleotides together is a phosphodiester bond. How is this bond formed? It's important you know it. A phosphodiester bond is going to be formed between, so if I'm trying to sketch something here, I have a sugar here that has a base and has a phosphate. I have another sugar that has a base and has a phosphate. So the bond is always between the number 3, the OH, listen very carefully, the hydroxyl group on number 3', which was on the ribose, with the phosphate on number 5. So you always be having a bond between the hydroxyl group on the number 3', which was found in the ribose of the previous nucleotide. Understand something? The hydroxyl group, that is OH group, the OH group on number 3' of a sugar in a particular nucleotide. So you have the OH group on number 3' of that sugar linking with the phosphate group on number 5' of the sugar in another person. So that if I'm linking them, you always be having 3' to OH from 3' to phosphate that was on number 5. OH from 3' to phosphate. So we keep linking, and those bonds are called phosphodiester bond. Now, with the way that bond is formed, you observe that you always have a free 5' end at one side, and you always be having a free 3' end. That's why you observe that anytime you're dealing with RNA and a DNA strand, there will always be a 3' end and a 5' end. The 3' end is where you're having the free hydroxyl group. The 5' end is where you're having your free phosphate group. But remember, there's always a link, and that's a phosphodiester bond. Now, just to add something to you, in DNA is double-stranded. So unlike RNA, which is single-stranded, at least remember in their strands, we'll have 3', 5'. And what is always linking them is a phosphodiester. But in DNA, who is double-stranded, what will link the nitrogenous base of one strand with the nitrogenous base of another strand is going to be another type. In DNA, remember DNA had 4 nitrogenous bases that we care about the most: adenine, guanine, cytosine. But we'll now be having in DNA what? Thiamine, not uracil. Now the link is always adenine to thiamine. That is ATGC. Adenine to thiamine, then guanine to cytosine. So usually the hydrogen bond between adenine and thiamine will be 2 hydrogen bonds. But if I'm linking guanine and cytosine, it will be 3 hydrogen bonds. All right, so that is all for the chemistry of nucleic acids. I am certain that you understood, but if for any reason you are still confused, just rewatch the video and I'm certain this will not be a problem again. So please do well to subscribe to this video. If you are new here, please subscribe. We have wonderful content that will help make your medical journey very easy. So subscribe to this YouTube channel. Ensure you like this video. Ensure you share it to as many people as possible. If you have a question, do well to type it out in the comment section until we come to you again talking about metabolism of nucleic acid. Thank you so much for joining us today. Until next time.